Okay, well, let's get started again. I was about to talk about secretory protein synthesis, and we talked about this a little bit earlier, so I'll go through kind of fast. But there's the nucleus. The DNA is there. It gives the code to the messenger RNA, and that goes down here, attaches to a ribosome. And this is the so-called rough endoplasmic reticulum. And here's a blow-up diagram of it. So the RNA gives the code to the ribosome, and the ribosome then assembles the chain of amino acids, the polypeptide chain. And here's then I'll show you another blow-up of this, but the point is that then that goes into the Golgi apparatus, and then eventually it's secreted to the outside of the cell. And here's a secretory pathway, and then there's also an unregulated. There's a regulated and an unregulated release pathway. And here's this is also the endoplasmic reticulum. This is the ribosome, and you can see the chain of amino acids is coming in here, and then it forms the disulfide bonds here and forms the ternary structure of the molecule. So it's very important that the cysteines in this chain are not oxidized until they get in here, and then this is a more oxidizing environment compared to the cytosol out here. And then the disulfide bonds can form, and they're chaperone molecules that make sure each one gets the right partner, and then that makes the right structure for the molecule. And then when it goes out, it will be recognized by the receptor because it has the right shape. And so those disulfide bonds are very important for that. So, so what happens here when glutathione is depleted is that these, I already talked about this export to the outside. They form the chain of amino acids, and they go in the endoplasmic reticulum. And there's the cysteine, and then they form disulfide bonds to give you the tertiary structure. Now, if it becomes too oxidizing, the cysteine will react with each other to form cysteine. And then when you do that too early, you can't make the right structure of the molecule. And in the case of ACTH, for example, it causes the molecule to go to the unregulated secretory pathway. And then some of them are also released in a misfolded shape so that when they go to the receptor doesn't recognize them. So causes a lot of problems with this category of proteins. So, so I would suggest that the reason that we don't have several of the proteins at high enough amounts is because of this mechanism, and also because this causes the HPA axis to be dysregulated, so that people don't have a high cortisol in the morning and then go down like they're supposed to. So glutathione then depletion is responsible for a lot of the endocrine abnormalities that you see in chronic fatigue syndrome. I might mention oxytocin is another molecule that's affected by this, and there are some others as well. Okay, this just gives an example of this molecule POMC, pro-opiomelanocortin. That's a molecule that contains ACTH. It also contains melanocyte stimulating hormone, and it contains endorphins. So this is kind of the pro-hormone, and it's made in a big chain, and then later on it's chopped up into pieces, and ACTH is one of those pieces. Well, here's the key thing. You see there's a hook here, a loop on the end of this molecule, and it's stabilized by disulfide bonds. So if glutathione is depleted, it messes this up, okay? It doesn't work right. And so now this was the signaling hook here that tells the cell that it's supposed to go to the regulated pathway. But if that hook is not there, then it doesn't go there, okay? And here's the HPA axis. It's actually, I've got HHPA because I put hippocampus on here as well. But you have the hippocampus, and then you have the hypothalamus, the pituitary, and then here are the adrenal glands. And you know there's the feedback system here that's supposed to regulate the cortisol output. But the problem is with the ACTH, it's not made properly, and so that's what messes up the regulation. Okay, now what about adrenal fatigue? and I know some of you are interested in that here. In my opinion, the cause of adrenal fatigue and chronic fatigue syndrome is usually not in the adrenal glands themselves. Now, in some cases it is, but usually not. But the problem is in the ACTH up in the pituitary, the mechanism we talked about. And that causes this problem I've already discussed here. And the other thing is the hippocampus is damaged by high levels of cortisol in the early part of the disorder. Remember I said cortisol goes high first. Well, the hippocampus has more cortisol receptors than any other part of the brain. And so this high cortisol likely damages the hippocampus. And that's probably also a contributor to the problems in the HPA axis. And how you fix that, I'm not quite sure, but I think it's a factor. Well, what about the so-called mild diabetes insipidus in ME/CFS? Now, you know, there are two kinds of diabetes. One is mellitus, which is the blood sugar, and this is the other one, diabetes insipidus. This one causes a high daily urine volume and constant thirst. And so it's caused by low secretion of antidiuretic hormone, also called vasopressin. And that hormone normally has 2 cysteine residues, so that's affected by glutathione depletion. And so I suggest that the reason for the problems here is that glutathione is depleted in the hypothalamus, and then that lowers the production of antidiuretic hormone. Then the kidneys dump more water into the urine, the blood goes low in water, you have hypovolemia, and the person is very thirsty. They keep drinking water, but they can't keep up with the loss, so they're always running at too low of a blood volume, always running hypovolemic. Well, what about the brain problems? Well, the two organs that are the most difficult to understand in chronic fatigue syndrome, in my opinion, are the brain and the gut. And the reason is there are too many things that come back and affect those. And there are too many feedback loops and interactions that come back. So these are just some of the things, and there are probably other things that affect the brain also. Glutathione depletion causes excitotoxicity, and the way that happens is, you know, glutamate is the main excitatory neurotransmitter in the brain. And when glutamate levels go too high in the synapses, it stimulates the NMDA receptors too much, and that produces excitotoxicity, which involves things like anxiety, nervousness, and insomnia. Well, when glutathione is depleted, it affects the astrocytes, and their mitochondria do not make enough ATP, so they have trouble importing the glutamate and converting it to glutamine, which they're supposed to do, and that leaves more glutamate in the synapses, and that causes the excitotoxicity. Methylation deficit leads to a dysfunction of the dopamine D4 receptors, and this has been worked on by Richard Deeth, a professor in Boston. He's been working a lot in autism and has the same problem there. But this affects attention. This dopamine D4 receptor is what allows you to concentrate your attention on something and hold it. And when people are unable to do that, it suggests that there's a problem with the dopamine D4 receptors. Also, myelin. And we have some evidence that myelin is not in good repair in this disorder because of the low processing speed and the slow reaction time. And also in magnetic resonance imaging of the brain for many years, there have been these white spots in the white matter of the brain, and it looks like that the reason for that is that this is damage to the myelin. The white matter is myelin in the brain, so I think that's what causes that.
Are these spots the same you see in MS?
I think so, but well, I shouldn't say that. These are sort of punctuate little spots. I think in MS you see a more damage than you do in this. But there is damage to myelin in both of these disorders, it looks like. Okay, well, the mitochondria in the brain are also dysfunctional, just like they are in the muscles and other places. And that causes elevated lactate because the mitochondria are not able to take the pyruvate and run it through the Krebs cycle and all of that. So, and that's been observed in magnetic resonance spectroscopy. So it looks like that's probably valid. More brain problems. The neurotransmitters, both the synthesis and the metabolism depend on methylation, and that's got a problem. The blood flow to the brain is lowered, and part of that is because of low total blood volume due to the diabetes insipidus that we just talked about. So there's a little feedback here. Diastolic dysfunction of the heart, which is due to mitochondrial problems in the heart muscle cells. And then defective control of blood circulation, which is due to problems with the sympathetic nervous system. So all of these things come together to affect the blood flow to the brain. And then finally, there can be infections, and we've seen that in some of the autopsies, and also using SPECT scanning of the brain. You can see that there's vasculitis, and thought to be due to infections, and that's because the glutathione depletion and folate draining impact the cell-mediated immunity. So things like viruses are difficult to combat.
The defective control of blood circulation can also be due to low functioning mitochondria, because they don't have enough energy to regulate blood flow, and then you will get ischemic pain. Could be one thing within fibromyalgia.
Okay. Makes sense to me. Okay, what about the immune system? Well, for many years, we've known that the natural killer cells and more recently the CD8 T cells have low cytotoxicity. They're not able to kill infected cells as well as they should be able to. And that's caused by low perforin secretion. This is really interesting here. Okay. And as I said earlier, I think that's due to glutathione depletion in these cells, which, because they have 20 cysteines, and so it's hard to make a good perforin molecule. The cell-mediated immune response is inhibited by the glutathione depletion, and so there's a shift to the Th2 immune response, and so that means that you're making more of the antibodies, but you're not able to do the cell-mediated immunity that's important for viruses and fungi, yeast, and intracellular bacteria like mycoplasma or chlamydia, rickettsia, those kind of things. So those happen to be the infections we see, and the reason we do is because that part of the immune system is suppressed, and so those are able to reactivate or get out of control. And we also see persistent interferon-driven RNase L activation. This is supposed to be sort of a stopgap measure. When you get a viral infection, the interferon secretion rises, and this is one of the systems that then activates, and it starts chopping up RNA, and that's supposed to slow down the virus. And that's supposed to be a temporary thing until the cell-mediated immunity gets up to speed, and then they come roaring in like the cavalry, and they take over and kill the bad guys, okay? Well, the cavalry never arrives in chronic fatigue syndrome, and so this stays activated. It keeps chewing up RNA, and then also the RNA itself gets chopped up and forms a low molecular weight version of it, which is unregulated. That's due to activation of calpain, and that happens, again, because of glutathione depletion. So you can see these things just sort of avalanche on you, and they cause more and more troubles. It's been observed that one of the tests immunologists do is they put in mitogens, and that's supposed to cause the B cells or the T cells to rapidly multiply, and you can see if they're very vigorous and ready to go on the attack. Well, they don't multiply in this disorder. Well, why is that? Well, because of folate deficiency. You need DNA to make new cells, and that's been drained out, so you can't respond. So this is one of the reasons why the immune system just doesn't respond well to infections. Pro-inflammatory cytokines, those tend to be high, and again, that's because of activation of the immune system. The immune system sees the bad guys. They know they're there, and they're trying to fight them, and they're putting out cytokines as signals to each other to, you do this, you do that, but they're unable to do it. So they keep sending these signals out, and you keep seeing high cytokines, but nothing good happens from it, okay? The messenger is sent, but they're unable to act on the message. Okay, viruses that normally remain latent in most of the population reactivate. Now, probably all of us here have certain herpes family viruses and other viruses that are in our bodies, and they're latent. They just sit there. Unless you get really under a lot of stress, your immune system goes down, or you get too much ultraviolet light on your face or whatever, they just sit there and you never hear from them. But in this situation, because the immune system is dysfunctional, those things reactivate. And you see Epstein-Barr virus, you see the herpes simplex 1, you see the HHV6, you see the cytomegalovirus, you see all these guys that usually are quiet and latent, they're coming up. So the ones we see are the ones would be caused by that. Well, then we come to the digestive system. Probably everybody's familiar with the digestive system. I just have a nice picture here to switch the subject. But there are problems in pretty much each part of the digestive system in chronic fatigue syndrome. So what causes those? Well, glutathione depletion causes low ATP production in the mitochondria of the parietal cells. Now, the parietal cells are the cells in the stomach that make the acid. They pump hydrogen ions into the stomach lumen to raise the hydrochloric acid. Well, those cells have to work against a big concentration gradient because the pH in the cell itself is around 7. The pH in the stomach, when it's running at the right acid concentration, can be 2 or some number like that. It's very, very high concentration of hydrogen. So these cells have to pump against that concentration gradient. Because of that, they have a lot of mitochondria. They need a lot of ATP. So when glutathione is depleted, mitochondria are affected. They can't make the ATP. Stomach acid goes down, okay? When stomach acid goes down, it does a lot of bad things for you. First of all, you get gastroesophageal reflux. Now, unfortunately, a lot of times this is interpreted as too much acid, but in a lot of cases, it's really not enough acid because the acid is necessary to signal the pyloric sphincter to open so that the food and the acid will go down into the intestine. And that doesn't happen if acid is too low, so it comes back up and starts burning the esophagus. So that's one thing that happens. And that's counterintuitive, and a lot of doctors will just give you a proton pump inhibitor and say you had too much acid, and they'll shut it down even more. Well, that causes the rest of these problems to happen then, see? Normally, when you eat food, especially fresh food, you have yeast and bacteria coming in, and the acid kills it in the stomach before it gets into the intestine. But if you don't have enough acid, those bugs go on down into your gut, and some of them are not very friendly, and you have dysbiosis forming. You get poor absorption of B12 because the first thing that happens on B12 absorption is you have to separate the B12 from the proteins coming in with your diet, and if you don't have acid, that won't happen. So you can't free the B12 in order to bind it with intrinsic factor and absorb it and go down the whole thing. So you go low in B12. Conversion of pepsinogen to pepsin, which is the first step in protein digestion, that takes acid. And if you have no acid, you can't do that. Signaling to the pancreas and the gallbladder to release digestive enzymes and bile, which starts the digestion. Well, if you don't have the acid there, you don't send a good signal. So now the food comes in, and the digestive enzymes and bile are not there to help digest it. So meanwhile, you've got all these bugs that you brought in. I don't know how that happened. I guess that's to keep me awake here or something. Yeah, so anyway, the combination there is just awful, okay? You've just set yourself up for all these bad guys in there, and then you deplete your own digestion so that you leave that food in your gut, which feeds these bugs, and on you go, You end up getting dysbiosis, you get leaky gut syndrome, and all the rest of the unpleasant things. Well, here's more. Lowered bile volume. Glutathione contributes to the secretion of bile, and so it's low. We just talked about that. Methylation block lowers the production of acetylcholine and serotonin. Now, those are the neurotransmitters that control the motility in the gut. So now that's messed up, and the food doesn't move through as fast as it's supposed to. That gives the bugs more time to chew on it, okay? So just, if you were trying to do this on purpose, you couldn't do a worse job than happens naturally here. Also now, normally glutathione is in the gut to bind toxins and take care of oxidative stress. Well, now that doesn't happen. So, and then you have depletion of the IgA, and probably that's due to the cortisol problem. And a lack of folate inhibits the making of new cells to line the gut. Now, the cells lining the gut have a short lifetime, and you need to replace those. And if you have low folate, then you can't make the DNA to do that. So now it's hard to repair the gut when you get the leaky gut. Well, anyway, leaky gut, and then that lets proteins into the blood, and the immune system responds to that, and now you have multiple food sensitivities, and the immune system is constantly activated trying to take care of these guys that have leaked in. Okay, so you can see what a terrible situation it is. Mercury, okay, mercury. I think those of you who are here know a lot more about mercury than I do, but I think it contributes both to the onset and also it builds up after the onset. Now, the contribution to the onset, well, if you have a big mercury load, you're going to contribute to the depletion of glutathione because glutathione normally takes out mercury. So that's one of the contributors to depleting glutathione. And then once you have onset, you don't have enough glutathione to take out the mercury, so then it can build up. And that ends up blocking enzymes and causing a lot of other problems, even including the pathway for glutathione synthesis. So this is a potential for another vicious circle mechanism. And you can see that's no fun either. Okay, thyroiditis, Hashimoto's. Now, what I've done is adopted this model from Duthoit here, okay? And there may be more going on in the thyroid than I'm talking about here, but I think this is one thing. This gentleman is from Sweden, I believe, right? Wickland. And he found that Hashimoto's is something like 40% of the patients had this Hashimoto's thyroiditis. And Byron Hyde in Canada has confirmed that it's very high. He sees a lot of it. Now, the thyroid cells normally make hydrogen peroxide as part of the process for making the thyroid hormones. They manufacture it. I'm going to probably move on to the slide that shows the picture there, and then I'll try to cover these things as I get there. Okay, so here's the thyroid gland, and or the cells from the thyroid. And the thing I want to mention here is this enzyme right here produces hydrogen peroxide, and it's pushed outside the cell. It's exported, okay? And then the reactions to make the thyroid hormones using hydrogen peroxide occur outside the cell. And that keeps the cell from being exposed to all this hydrogen peroxide. However, some of it leaks back in, okay? Now, normally, glutathione is right here to convert that to water and take care of it. But if your glutathione goes down, now you got this hydrogen peroxide here, and it starts damaging things. And according to Duthoit's mechanism, it can attack proteins and cause them to look like foreign molecules to the immune system, and that causes then the immune system to respond, and you have an autoimmune attack, and that's Hashimoto's. So if that's true, if you get glutathione back up again, and your thyroid isn't too badly damaged, you should be able to pull out of this. And we have had one or two people who have said to me that's happened to them. So, but I don't have a lot of data on that yet. Okay, what about the electromagnetic hypersensitivity? Well, there's several mechanisms been suggested for that, and I don't disagree with those, but I want to suggest another one here that I think is present in chronic fatigue syndrome. Normally, myelin is electrical insulation for the axons, and over time, the myelin becomes damaged in chronic fatigue syndrome. Well, in any case, it becomes damaged, and it's normally repaired. But you need at least 3 of the substances you need to make myelin require methylation. Okay, that includes myelin basic protein, phosphatidylcholine, and choline plasmalogen. All of those require methylation to be made. If your methylation is not working well, then you have trouble making those things, and then I'm suggesting the myelin falls into disrepair. So now you don't have the normal insulation on your axons, and when you're walking around in the environment with all this electromagnetic radiation coming from every direction, it's inducing currents in your body. Usually those currents don't flow through your nerves, but when your nerves are not insulated, those currents flow into your nerves, and now you have neurological symptoms. So I'm suggesting that that's the mechanism in chronic fatigue syndrome. Now, you know, I know that Henry Lai has also suggested that there's a mechanism to make oxidative stress. And if that's true, I mean, that falls right into our laps again. We have glutathione depletion, you make more oxidative stress, you can't take care of it, it's another problem. So there could be other mechanisms, but I'm suggesting this is one of them. Well, there's the myelin. This is a neuron, it has its axon, and then that's the myelin insulation. Okay, testing. Now I'm going to start talking about how would you test, go ahead.
You don't have looked at the vitamin B12 and myelin repair.
Well, there could be another mechanism for how vitamin B12 is involved in myelin, but one of them is that it stimulates methylation. It supports methylation. And so when B12 goes down, methylation goes down, and you can't make these three things that require methylation, so you can't repair myelin. There could be another mechanism, you know. I mean, we know that B12 is involved in myelin, but I'm suggesting that's how it's involved. You know, there may be other ways.
I am electrosensitive, and my feelings is that vitamin B12 helps me to who could be out in the society, and my theory is that it helps to repair myelin.
Helps repair myelin. Okay, I think you're right. Yeah, I think you're right. Yeah. And when we get to the treatment part, B12 is part of the treatment. Okay, so yeah.
Yes, I know.
Okay, now I want to mention here, this is the panel that I prefer. If a person's only going to run one test, this is a good panel, and it's offered by the European Laboratory of Nutrients in the Netherlands, and also by their branch in the U.S. And this is the commercial part here now. Here's the commercial.
Will be introduced.
Will be, okay. So it's going to be possible to order this through ScanLab, and he's working on that. So there's your representative for Sweden, okay? Now these are the things that are on this panel, and you can see a couple of them are involved with glutathione, and these here are involved with the methylation cycle, these three actually, and then the rest of these are involved with the folate metabolism. So this panel evaluates all three of those areas.
Is plasma the best way to measure the level of glutathione?
Yes, and the other thing about that is we really want to know reduced glutathione, okay? And if you take a blood sample out of the body, it's going to start oxidizing, okay? So these labs here are the only ones I know of who have figured out which enzyme blockers to put in the sample vial. So when you take the blood sample, it will preserve the reduced glutathione, so you'll get a good measurement. And I don't know of any others who can do that. And of course, it's proprietary which ones they use, right? It's a commercial advantage to be able to do that. But we have found that the plasma measurement is a better one to make than whole blood or red blood cells. And there are other labs that offer total glutathione in the red cell. That's the easiest one to do. But you know, it really matters whether it's really reduced, which is the active form, or whether it's oxidized. So you really want to know that. So anyway, this one is a good diagnostic for whether this model really is involved in a given case. And also, it gives you baseline data so that if you do some treatment, you can see later, run this panel again, you can see whether you've had some improvement. Okay, now what about, is there other testing, indirect testing that you can use? Yes, you can do urine organic acids testing. For example, Genova Diagnostics Lab offers that, and Scan Lab here is very happy to have your business on that too. They represent Genova Diagnostics. So on that panel, there are lots of things, but these three are particularly important. One is the methylmalonic acid. This is the FIGLU, and that's the pyroglutamic. And when MMA and FIGLU are both elevated, when they're both high, that's pretty good. Okay. That's a pretty good indication that you have a problem in the methylation cycle, because one of those is looking at B12 dysfunction or a functional deficiency, and the other is looking at a problem with folate. So when they're both there, it's a pretty good chance that you have the problem, but it is indirect. And then pyroglutamic, if it's either too high or too low, then likely you have a glutathione problem. And typically in a chronic fatigue syndrome person, you see all of those, okay? You see them all together. So that's a pretty good indication. Okay, now I mentioned earlier Dr. Yasko, and she evaluates 30 different polymorphisms in these genes. Now, some of them have more than one polymorphism, but these are the genes. And she, as I mentioned, these have not been statistically analyzed to find out, are these more prevalent in a person with chronic fatigue syndrome than in the general population? There's been some informal studies, and we know that 2 or 3 of them really are. For example, this one here, which is angiotensin converting enzyme. That one has a deletion to it, which causes it actually to be more active. And we see this in almost every person with chronic fatigue syndrome. There are some who don't have it, but most of them do. And so it's very prevalent. And the effect of that is to cause angiotensin 1 to be converted to angiotensin 2 at a higher rate than normal, and that tends to raise aldosterone. So that affects the adrenals. Well, normally in chronic fatigue syndrome, there have been 2 studies now, and they find aldosterone is low, not high, okay? So there's more that goes on after this, but it could be that early on, aldosterone is actually running high in these people. Usually people don't measure it until they're sick, and then, it's low, so you don't always know. Yeah.
Yeah, we work very close with Dr. Yasko as well.
Okay, there's another.
First time we cannot.
Okay. Well, the presence of these SNPs or polymorphisms shows tendencies toward problems. They don't tell you exactly what's going on, and that's why I like the biochemical panel I talked about before, the methylation pathways panel, because it tells you exactly what you have. This tells you what you tend to have. And if you have a lot of these polymorphisms, it explains why you're tending toward a methylation cycle problem. And Dr. Yasko has a rather elaborate treatment program, and that's described in her book called Autism Pathways to Recovery. And she also has a website, and she's written this book called Genetic Bypass. Okay, so these books talk about the effect of polymorphisms in those enzymes on the methylation cycle and related pathways. And then she has she's a naturopath, so she uses all supplements for treatment, and she has a very large number of supplements. Some of them are specifically designed by her organization. And if you have a certain polymorphism, she says, aha, you need this supplement or these 5 supplements. So it tends to be a rather complex program. It tends to be expensive, involves a lot of testing, and sometimes people end up taking 50 supplements a day, So some people are willing to do that, and I think there's some benefits to it, but what I found in working with chronic fatigue syndrome people is, number 1, a lot of them didn't have any money, and number 2, they didn't have much brain power either because they were sick. So that's why I developed a more simplified version of her program, and that's what I recommend people try first. Now, sometimes that doesn't do it, and in that case, they need to look further, and if they're willing to and are able to, then they can start to look at some of these things and figure out, well, maybe I should add this or that to try to improve my chances, Okay, so So what treatment is suggested by this hypothesis? Well, you might think, well, raise glutathione. That sounds like a good idea. Well, I tried that for 5 years, okay? We put glutathione into every body orifice and everything you can imagine, and it did help some of the people, but only as long as you kept doing it. If you stopped, everything went back the way it was again, okay? It wasn't a permanent fix. So in 2004, I wrote a paper, poster paper for the conference there, and said, you know, glutathione helps temporarily, but it won't help permanently, and I don't know why. I don't know why it doesn't work, but there's something going on. There's a vicious circle somewhere. 2 months later, Jill James, who works in autism research, published a paper, and lo and behold, in autism, they also had low glutathione, but they had a problem in the methylation cycle as well. And when they treated the methylation problem, glutathione came up by itself. And I said, aha, this is it. We have the same biochemical problems that you do. So I told the people, you know, you need to start doing autism treatment for chronic fatigue syndrome. And so people started doing that, and it was helping. So that's how we kind of got going. But Jill James was the one who originally came up with that. So anyway, this treatment then would be to support methionine synthase with B12 together with active folate in order to lift the methylation cycle block. Now, that combination is the key to the treatment. For many years, people have used high-dose B12, and it helps the people some, but it's not permanent. But if you combine that with active folate, and I'll show you which one I'm talking about, that combination lifts this block, raises glutathione, and you see some permanent benefit. So that's the key. And it took us a long time to figure that out. Okay, and then you also might have to add other vitamins and minerals and amino acids because these people tend to be deficient in a lot of things. They've been ill for a long time, their digestion's not so hot, so they kind of go low in vitamins, minerals, amino acids. So you have to kind of build those things too. Now, just want to review a little history here of previously tested treatments with B12 or folate or both. And I think we have a representative of this study here, right? This is Professor Reglan et al. And here, well, I should start at the beginning. This is Les Simpson from New Zealand. And he was probably the first guy who said, use B12 in chronic fatigue syndrome. And he was looking at the red blood cell shape with his electron microscope. And he found they were distorted and improper shape. But if he gave B12, it helped him. And he didn't know why. Well, now I think we do know why. It ends up helping with the oxidative stress, which then causes the damage to the red cells to stop, and they have the right shape. Well, then Charles Lapp and Paul Cheney in '93 got the idea that maybe they should give high-dose B12. So they kept bringing it up, and they found that a dose between 2 and 2.5 milligrams, which is very high compared to the RDA for B12, which is a few micrograms in people, they found that they saw symptom improvement if they went that high. Well, then Professor Reglan et al. Did a study in '98, and they used 1 milligram, which is lower than Cheney and Lapp here, for at least 3 months, and they found that this treatment was significantly more beneficial if the patient did not have this MTHFR SNP. Now, I read that paper when it came out, and I didn't understand the significance of that, and no one else did either, as far as I know. But there's the clue right there, okay? You got to have folate with it, okay? The people who do have that, are not able to make methylfolate very well. So you need B12 plus folate, and you need to have it at this dose, okay? So we were all kind of shooting around it, but we didn't quite understand what was happening. Well, then Charlie Lapp was up to 3 milligrams, and he had improvement in most of the people. Well, then not too long ago.
Was it every second or third day, 3 milligram, or what does it say?
Every 2 to 3 days is what he means.
Intramuscular.
Intermuscular every 2 or 3 days.
Uh-huh.
but he wasn't using folate, only B12, okay? Now, here we had a study not too long ago, folate alone, and this was folinic. Now, that's a huge dose. You know, the RDA for folate is 400 micrograms or 800 micrograms if you're pregnant, okay? And this is 15 milligrams, huge dose. But he saw 81% of them had some improvement. So, but he didn't give B12 with it, okay? Just folate. Okay, now, here's a study in '89 where they did give B12 and folate together, but look at the dose, 220 micrograms, and you need something like 2 milligrams. So his dose was too low, and he didn't see anything very interesting. His folic acid was pretty good, but he used folic acid and not the active forms. So we now know that B12 dosage was not high enough here, and that other forms of B12 and folate are better than what he was using. He was using cyanocobalamin. You don't want to use cyanocobalamin if you're going to high doses because of the cyanide, so it's better to use other forms. And then Marty Paul, in 2001, recommended hydroxycobalamin to scavenge nitric oxide. He has a different theory for chronic fatigue syndrome than I do. And he also adding methylfolate to scavenge peroxynitrite. And so far, I don't think he's actually done that and reported any results, but he's recommended that. He said he was going to do that. Unfortunately, his wife died in July, and I think he's, you know, still reeling from that experience. She had cancer, so. But anyway, eventually I think Marty is going to modify his protocol and add these things, and we'll see if he gets results. Okay, so what protocols are in use now to lift the methylation cycle block? This is the one I've suggested, but it's not the only one out there that's being used. The autism people have their own protocols. Amy Yasko has hers. There's a doctor in Maryland, USA, who uses folic acid and hydroxycobalamin under the tongue, very high doses of folic acid. Here's a guy, Fred Davis, who's on the internet. He recommends very high doses of methylcobalamin along with methylfolate. And I have some differences with Fred. I think he has genetic issues that are personal to himself, and he's found that this works for him. But with that experience of n equals 1 himself, he now says everyone should do this, and I don't think that's the right way to look at it. But he has helped a lot of people. So, you know, I think there are other people probably who have some of those same issues. But the difficulty with using high-dose methyl B12 along with high-dose methyl folate is that you overdrive the methylation cycle. And when you do that, there's not enough flow of homocysteine down to make glutathione. So you're doing great on methylation, but you're not raising glutathione. You have to let the cell control that, let it get back in control so it controls how much is going here and how much is going there. And that's the problem with going too high. Now there's a pharmaceutical company in Louisiana, USA, which is licensed by Merck to use methylfolate. Merck has the patent on making methylfolate, and so they've licensed PamLab to produce medical foods, and these are their foods here: seroflonac, metanx, and depletion. Depletion is pure methylfolate. This one has methylcobalamin, methylfolate, and N-acetylcysteine, or NAC, and this one has some B6 in it, in addition to methylfolate and methylcobalamin. So that one is used, it's recommended for treating Alzheimer's or pre-Alzheimer's. This one is recommended for treating high homocysteine, and this one is used for depression. Well, there are doctors now using them off-label to treat chronic fatigue syndrome, and probably the one most commonly used is this one here, and the patients are saying it's helpful to them. I think the dosages are probably not optimum for chronic fatigue syndrome, but they have the right stuff in there, and it is helping some of them. Okay, so they all have forms of B12, and they have forms of folate. And the B12 is very large dosages compared to the RDA, and the folate doesn't have to be that high, relatively. So anyway, this is just going back to that diagram we saw before. Here's methionine synthase. There's the B12. There's the methylfolate. And the reason you have to give both together is it needs both of those to get going again. So this is the treatment I've suggested, and it's based on Amy Yasko's treatment, but I've extracted just the essential part of it and not all of it. So it's hydroxycobalamin, and again, we're at the dose that Cheney and Lapp found was effective, and that's the same thing they're using in autism. So it's very interesting that the same dosage. Methylfolate and folinic acid, and the sum of those is a little over 400 micrograms, which is sort of comparable to the RDA for folate, total RDA for folate. So I find you don't have to go that high in folate. I mean, that's a good number. You don't have to go way above the RDA. But you do have to go high for the B12. And then we use this multi here, which is a multivitamin, multi-mineral that Yasko has developed. And it has the vitamins, it has the minerals, and it also has some other supplements that help the methylation cycle, like methionine and N-acetylcysteine and trimethylglycine. There's some special ones in there in addition. So we use that one. Some people say, well, you know, it has a very large number of things, but I'm sensitive to that one there, so I can't use the pill. You know, one of them knocks me out, and that's the problem with the multiple like this. If you're sensitive to any one ingredient, you can't take the thing, and then you have to start with something else. So some people go for other multis, and like the Thorne Basic Nutrients is one that they use. And then the last one there, taken off. Okay, bye.
Sorry, take a break.
Okay. The last one there is a phosphatidylserine complex, and that has all of the phospholipids. It has phosphatidylcholine, phosphatidylethanolamine, so on. And that one does a variety of things. It supports the membranes, which have been damaged by oxidative stress, and it also helps to supply that BHMT pathway in the liver and kidneys, which helps to make S-adenosyl methionine, which can help this other pathway. So that one kind of does double duty. Now, some people do not want to take that because it tends to lower cortisol, and they say, my cortisol is already too low. I don't want to do that anymore. So for that, I've said, okay, use lecithin instead. And that doesn't have the phosphatidylserine at such high levels, but it still supports the membranes. So it's more phosphatidylcholine. Okay, how are we doing for time? Do we need to take a break or?